Okay, so in this video we shall discuss certain basic concepts regarding drug development and discovery. Now, the earliest stage in the development of a new drug begins either with the isolation and purification of a new chemical compound or with the screening or testing of existing compounds for potential use as drugs. Now, the discovery of drugs such as quinine, atropine, digoxin, drugs of these nature were mostly discovered by the first process, the isolation and purification of chemical compounds. Now, in the year 1824, alkaloids were extracted from the bark of a plant, Cinchona officinalis. Now, these included cinchonine, cinchonidine, quinine, and quinidine. Now, of these, quinine was regarded as the mainstay or standard of care in the treatment of malaria for many decades. This was of course until the introduction of artemisinin and combination therapies. Now, if you look up the process in the discovery of artemisinin drugs, they too have a similar history. Nowadays, however, many drugs are discovered by the latter method, the screening or testing of existing compounds for drug-like properties. Now, this particular method is employed by large pharmaceutical industries with huge research and development programs. They have got libraries, which we call compound libraries, or indeed chemical libraries. A compound library consists of a very large collection of real chemicals or virtual chemicals. And every day, up to 100,000, up to 1,00,000 compounds are tested for drug-like properties. And this is made possible by the use of highly sophisticated machinery, automation, robotics, and computer software. This process is what we call high-throughput screening. Now, the screening may be done either by testing these chemicals in the lab, which we call in vitro studies, or using computer simulation models, which we call in silico studies. Now, if any such compound shows promise for a drug-like property, it is identified by its IUPAC name and it is investigated further. Now, if the findings are truly encouraging, then this compound may be introduced into animals to understand its effect in the biological environment. Now, these are what we call in vivo studies. Now, any study undertaken up to this point may be called a preclinical study. So, this particular arrow shows you the journey in the development of a drug. Now, till this particular point, up to in vivo studies, we have reached what we call preclinical studies. Now, every step of the way, whether it's an in vitro study or an in silico study or an in vivo study, the findings or the evidence generated by these studies needs to be published. By publish, we mean it should be made available in public domain, and the evidence should be submitted to the regulatory authority of the land. So that's the duty of the pharmaceutical industry, the pharmaceutical company that has come up with this drug. It needs to submit the evidence to the regulatory authority of the land. Now, the pharmaceutical company that is conducting the research is called the originator company or the innovator company. Now, in the US, the regulatory authority is the US FDA, and in India, it is the DCGI, or the Drug Controller General of India. So, if the regulatory authority is satisfied with the evidence submitted by the originator company regarding the new molecule that they are coming up with, then the compound may be introduced into human beings for testing. Now, at this stage, the compound that is being tested is called the investigational new drug, or the IND, and studies that involve the administration of the IND to human participants are called clinical trials. Clinical trials go through various phases. They go through 4 phases in general. Now, phase 1 studies are called first-in-human studies because that's the first time this drug is introduced into humans. Now, the point to note in phase 1 studies is that it is not patients that are being recruited for this study. It's healthy volunteers. Now, the number of study participants for this phase is around 10 to 100, and the evidence collected in phase 1 studies pertains to the safety and tolerability of the drug. Now, once phase 1 studies are over, the evidence generated needs to be published in journals and the data needs to be submitted to the regulatory authority of the land. If the regulatory authority permits, then the innovator company or the originator company may proceed to phase 2. Now, phase 2 studies are also known as first-in-patient studies. Around 50 to 500 patients may be recruited for phase 2 studies, and evidence is collected regarding the efficacy and the proper dosing of the drug. Again, the evidence collected from this study needs to be published and needs to be submitted to the regulatory authority of the land, and if they give their approval, then the innovator company or the originator company may proceed to phase 3, which is also known as multi-centric studies. So here the study may be conducted in multiple centers, possibly around the world. The patients may include numbers ranging from 500 to a few thousand, and the efficacy is confirmed in these large studies. Again, the evidence or data collected from this particular study needs to be published, put in public domain, and it should be submitted for approval to the regulatory authority of the land. If they approve, then the originator company may finally release their drug into the market. But that does not absolve them from responsibility of collecting data for the next 2 years, following which the drug enters the market. And this is what we call post-marketing surveillance or phase 4 studies. So thousands of patients will be included in this study, and additional real-world data is collected regarding the drug. And this includes things such as adverse effects, drug interactions, things of this nature. Yeah. So now if we look at this process, here are the preclinical studies and there are the clinical studies, phase 1 to phase 4. And after phase 3, the drug enters the market, following which there is post-marketing surveillance. Now, this entire process takes years, 10 to 12 years to undertake. It takes billions of US dollars. But during this entire period, during this period of 10 to 12 years, evidence, a lot of crucial evidence is gathered and documented about the drug. Now, this evidence includes information about what the body does to the drug and what the drug does to the body. Information is collected as to how the body absorbs, distributes, and metabolizes the drug with respect to time. And this is what we call pharmacokinetics. Information is also collected regarding how exactly the drug produces its responses in the body with respect to various doses or plasma concentrations. And this is what we call pharmacodynamics. Right. Okay, so now we move on to drug nomenclature. Now, during the preclinical phase of drug development, the compound is known by its IUPAC name, but IUPAC names are long, complicated, and horrendously difficult to memorize. So, during the R&D process, the investigational new drug gets named by the WHO. Now, the WHO assigns a name to this drug, and this name is called the International Non-Proprietary Name. So, for example, the International Non-Proprietary Name for the long IUPAC name that you saw in the previous slide would be something as simple as diazepam. So each international non-proprietary name is unique, it's the same all over the world, it's public property, and it is the name that we use for safe prescribing, for teaching, and so on. Now, on the other hand, the innovator company or the originator company, they have a right to name the drug which they have poured billions of dollars in to produce. So, the name given by the originator company or the innovator company is called the originator brand name or the innovator brand name. For example, the very first brand of diazepam to ever enter the market was called Valium. Now, for a certain period of time, a number of years, no other company is permitted by law to produce or market this particular drug. This is to give the innovator company or the originator company time to recoup the huge amount of time and money that they have invested into developing this drug. But once the patent expires, then any company can market this drug. Any company can produce and market this drug. Drugs of that nature are called generic drugs. Now, so that's a generic drug. Now, generic drugs may be named in two ways. One way is to not bother to give it any additional name whatsoever, just use the international non-proprietary name. This is how governments usually produce drugs. For example, if the government of India wanted to market diazepam tablets, they can very well do it under the name tab diazepam. However, if there is another company who wants to market their own brand of diazepam and they want to give it their own name, they can do that. They are permitted to do that. For example, there is a common brand name in India for diazepam called Campos. So that would be an example for a branded generic drug. So this was in brief regarding the process of drug development and discovery and a little bit about drug nomenclature.